This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1231, recorded on the 27th of June, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is currently 75 Fahrenheit, which my phone tells me is 24 Celsius, and very overcast outside. It's a nice break from big time heat earlier this week. So I'm pretty happy about that.
25C here and cloudy in Chelsea. Yeah, it's so different.
Mm-hmm.
My gosh, it was so hot. I walked all over the city on Wednesday.
Me too.
And it was really, were you in the city on Wednesday?
I was there Tuesday night and all day Wednesday. It was so hot.
It was hot, but it was so weird. I didn't sweat. I'm like, what's wrong with me? I'm not sweating at all. Did you sweat?
I did. Maybe I sweated enough for both of us.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I can't actually tell you about the weather because I can look at my phone. I probably ought to do that.
Ask the cat.
Yeah, well, the cat is going to be an issue here for a little while because he is a prisoner in my office, and he's not real happy about it.
Why
I'm just
can't he
going to
get
have
out?
to let him, because there's somebody out there cleaning the house,
Oh, I see.
okay? And he could get out, but that will be even a more complex environment than here. He's going to have to deal with the vacuum cleaner.
You're looking for the weather, is that right? Now you're looking for nothing because you're frozen again.
Oh, no.
It's going to be a long podcast. Also joining us from Montreal, Canada, Angela Mingarelli.
Hello, everyone. It is 78C. I think that's 25. Sorry, 70. 78F. 25-ish C. Wow, that was a mistake. And it was also extremely hot here earlier in the week. It was like 105 F, like 40 degrees. And with the humidity and everything, it felt like 110. It was crazy. I also sweat for all of us included. Like it was very, very, very
I never
hot.
did sweat very much, like beads of sweat kind of thing. I mean, the skin gets a little damp, but I remember walking around Manhattan and it's like, this is interesting. It's the way it
Maybe
is.
it was so hot it just evaporated
I'm
instantly.
just a cool cucumber. Cool as a cucumber. I don't know why that's a word because cucumbers are not necessarily cool, right? And we were talking about words before we came up with sketchy. We were talking about the tech of our recording.
That's
It's a little
pretty
sketchy.
I love the word sketchy, actually. I think it's a hilarious word. Sketchy, creepy. I love those words.
If you like these programs where we talk about science, real science, not lies, not misinformation,
Not sketchy science.
not sketchy science. Yeah, we have quick hosts on this show. Just like come up with stuff like that. It's amazing. You should. We'd love your support, financial support, because it takes money to produce these podcasts. I don't do it myself anymore. Those are the old days. Have people helping, and we don't do ads, so we'd love your support. Microbe.tv/contribute. We just had a visitor here, and he said, I want to contribute, and he pulled out a credit card. No, I don't have a credit card reader here. Wouldn't be any use for it because I don't sell anything, right? Microbe.tv/contribute. So you're there, I think you can use a credit card, or you can use PayPal, or you can use other things, or you can mail us a check. Of course, you can come here and give cash, too. Some people have done that. It's very unusual. But we don't keep cash at the incubator, so don't get any ideas, okay? You can come here and take stickers if you want. We have plenty of stickers, but no cash. Microbe.tv/contribute. ASV 2025 is coming up in just like a couple of weeks in the city of Montreal, where Angela might be. You're going to be there, right?
Will be.
That should be fun. What's the weather going to be? Really hot or nice?
Well, it's always hard to know like 2-3 weeks in advance, but normally July is actually pretty hot. It should
That's
be
okay.
like 25 at least, almost
you know
30
what?
C.
I'm not going to sweat it.
But yeah.
Do
You
you,
literally won't
Angela,
anyway.
do you have any official duties for the meeting?
Official duties, no. I'm trying to plan a little TWiV gathering potentially and one of the social events in the evening just with Kathy and one of the organizers to see if Brianne, Kathy, Vincent, and myself can make ourselves available in a specific area if people want to come, say hi or talk or whatever, in one of the evenings. So we will confirm. I'll confirm with Vincent by next week, and then we can say on TWiV so everybody knows where to find us.
That'd be cool.
Yeah.
And we'll push it out on social media.
Exactly.
I'll be
The
in
social
Oregon.
meetings are like, go pretty late.
It does go very late. The sessions
11.
go late, and then they have a mixer every night. Yeah.
Mm-hmm.
So you're going to be in Bend?
Yeah, Sun River, south of Bend.
Good.
South of Bend? South of Bend. Yep.
We're going to miss you, Rich.
I will miss you guys. I always enjoy ASV. Haven't been for a while.
It's our meeting.
Yeah, I know.
That's where the first live TWiV was.
Oh, wow.
I think. Maybe it was in Philadelphia, actually, but there are only 2 people in the audience, 1 person. So ASV, we actually had an audience. Okay, so it's Montreal, July 14th through 17th of this year. The website is asv.org/ASV2025. And 2 reminders from Kathy. One, remember that one of ASV's sustainability initiatives is to reuse the name badge holders. So you get these name badge holders, which are really big, kind of nerdy. They're nerd flags. See if you can find yours and put it with your suitcase. So find an old one. So
I cleaned out the top drawer of my desk the other day and found probably about 8 or 9 lanyards.
Yeah, you tend to keep them.
They accumulate, yeah.
Yeah, I happened across my name badge holder the other day going through my desk for something else and was like, oh, yes, this is important.
We have them in our lab. We use all of from our conferences or whatever. Everyone in our lab hangs them next to their bench. So we all have a million lanyards and like plastic things with our names. So
I was going to say, I keep mine.
Mm-hmm.
I don't know if you can see this. But
Yes.
Yeah, I've
Cute.
seen that picture before. That's good.
But this is only like 20. I throw them out periodically because there are too many.
It's the science version of the Olympics.
Yeah, except these are all bronze. Anyway, so bring your old badge. And also, email reminders have been going out with helpful things. If you missed those, many of them are on the homepage of the meeting, asv.org/ASV2025. Scroll down and read the announcements, the newest at the top. You can click on them, and they will expand. Okay? Then finally, standard registration for the meeting ends on June 30th. And after that, the rates go up. So you should, this is going to come out on the 29th. So you better hurry if you haven't done it already. I just want you to get the good deal, All right, we have a bunch of news items because, it's crazy these days. It's totally crazy. Did I save these as PDFs? I hope so. No, I didn't. Oh, darn. I had meant to do that. Where are they? Anyway, I will tell you, because I remember these. I do have the articles. First is a New York Times article, Cassidy in break with Kennedy calls for a vaccine meeting delay. I don't even have to look at the article because I know what happened. So this is Senator Cassidy of Louisiana, who is an MD, right? And he voted to confirm RFK Jr. in the Senate.
He was what most people thought would be the deciding vote on one of the committees that was doing some of the hearings for Kennedy.
And he took his time. It was suspenseful.
So he said he had talked to Kennedy, and Kennedy assured him that he would listen to Cassidy's advice about the ACIP, and he wouldn't take away any vaccines. And of course, he's gone and replaced ACIP and wants to take away vaccines and so forth. So Cassidy's a little upset. So he said, don't have this ACIP meeting, which took place already anyway, because they didn't pay any attention to Cassidy because that's the way this administration works. And the meeting happened Wednesday and Thursday, and we summarized what happened on Daniel's clinical update. So I won't go into that at all. But the people are just completely unqualified to be on this committee. I mean, this one lady got up and talked about thimerosal, which is only in the multi-dose flu vaccines. And she said, I'm speaking as a civilian. She had no expertise. She used to be head of this nonprofit that Kennedy founded, which is an anti-vaccine company. So people like that. And then this one guy who's the chair of the committee, Koldorfer, he's got a PhD, but he thinks we don't need experts. He said everyone should have an opinion. And this is the most stupid thing I've ever heard. Ask him if he would like somebody else to fly his airplane next time rather than an expert, right? Or somebody other than a surgeon. Let Rich Condon cut you open and take out your gallbladder. How about that?
That would not be a good
Well,
idea.
I think everyone can have an opinion, but I don't care about everyone's opinion. I care about well-reasoned, informed, factual evidence.
That's right. That's
You
right.
should go read the book, The Death of Expertise by
great.
Tom Nichols
Yep.
that you
Good
suggested, Vincent. You should go read that.
Yep.
Many insights.
Another New York Times article. Kennedy accuses International Vaccine Agency of ignoring science. All right, so there's an international vaccine agency called GAVI, Global something Vaccine Initiative. I forgot what the A stands for. GAVI. And so GAVI basically raises money, about $9 billion a year, and they buy vaccines for countries that can't afford them. All kinds of vaccines, including polio vaccine. And so last year, Biden agreed to give $1.2 billion to GAVI. And Kennedy said, nah, we're not going to give it to you because you don't pay any attention to vaccine safety, which is a lie. It's basically a lie. Of course, they pay attention to vaccine safety. They use vaccines that are WHO approved. And just another meddling for no good reason, right?
So it's not just that they're going to...
Sorry.
Oh, I was going to say, it's not like they're just going to hurt people's health here. They're also hurting people's health and vaccine availability around the world.
That was one of the themes of this. He's now reaching out to mess with vaccines elsewhere.
Global Alliance for Vaccines and Immunizations. Got it.
Alliance, very good. You must know that being on Brighton, right? Gavi.
No. Well, we talk about GAVI all the time, but, it's such an alphabet soup out there that I have to look these things up all the time. I even have trouble with, CEPI, what's that? Coalition for Epidemic Preparedness Innovation.
Intervention.
Preparedness and innovation.
And then I'm on a subcommittee that's this safety arm called SPEAC, SPEAC. I can't tell you what that means. It's okay.
Some of the acronyms spell a word, which is fun, and they're actually somewhat related, right? Okay, so that's that. And then the next article is in The Guardian, which you can get to after you get through all the advertising. Millions of children at risk as global vaccine rates fall. Decline blamed on health inequalities, COVID disruption, and soaring levels of misinformation and hesitancy. So it just goes through all the countries and talks about what's going on. Like measles vaccination has fallen in 100 of 204 countries since 2010. Coverage for diphtheria, tetanus, whooping cough, measles, polio, TB has declined in 21 of 36 high-income countries, etc. So it's a list of all the bad things that are happening. I mean, these are vaccine-preventable diseases, bless you.
Thank you.
Which just unacceptable. So it's a good article that details it. All right. And then the next one is a USA Today story. This is RFK Jr. grilled by Congress over health spending cuts, vaccine overhaul. So he was on June 24th. He went in front of Congress for a couple of hours. People are wanting to know what he's doing, cutting programs and all of this stuff. But the one thing I wanted to point out, he had this exchange with Frank Pallone, who's a Democrat representative from New Jersey. he said he was concerned about Kennedy's leadership. He says, the thing that disturbs me most is the anti-science agenda. And Pallone said, most scientists and medical professionals believe Kennedy's views on vaccines are dangerous and not supported by scientific evidence. I think people are going to die as a result of your actions and congressional Republican actions. And so Kennedy said, you know, you take millions of dollars from Big Pharma. You're the worst offender. You shouldn't be talking. And it's the lie. He just lied. He just said that because nobody's going to check it. But fortunately, the chair of the committee, who was a Republican, said, you have no evidence for that. Take it back. And he had to retract his comments. So at least that's good, right? He talked about ACIP and all this stuff. He said he fired ACIP members who had conflicts with the pharmaceutical industry. Another lie. They had no conflicts. They went through a conflict of interest exercise shortly before.
The whole thing is Orwellian.
Yeah,
Okay?
no,
It's unbelievable. War is peace, right?
Yeah. And finally, this one is very good. Let me see. Let me get the original article, which is in another folder. It's in my office hours folder. Bear with me one moment, folks. I got everything nicely. Cassidy. Here we go. Opinion piece. Okay. Oh, my sync is paused. Just a minute. I got to turn Dropbox back on. You know, everything is like.
It's going great. Except for science in the United States.
Everything is connected. Here we go. This is an opinion piece in the Times by Alan Francis, who's a psychiatrist, and he was chair of the American Psychiatric Association Task Force. And the title is Autism Rates Have Increased 60-fold. I played a role in that. He says, RFK is right. Autism rates have gone up in the last 30 years. But he's wrong. He's dead wrong about the causes. I should know because I'm partly responsible for the explosion in rates. The rapid rise in autism cases is not because of vaccines or environmental toxins, but rather the result of changes in the way it's defined and assessed, changes I helped put into place. And the rest of the article is all about what he did and how it's changed. Okay, so this is an expert who knows what he's talking about, and you know he explains. RFK has just brushed this off. He said, no, it's not about reporting. It's an environmental toxin because it doesn't fit his agenda, right?
This is a really good article. I really like it.
It's very good. It's an opinion, right? But it's fine. Yeah.
It's an opinion that's supported by facts and evidence.
Gee.
Imagine.
I know. It's unusual, right? All right. So that's our news for the week. There's even more, you know, at the last minute things come out, but that's enough. We'll cover them next time.
It's enough about RFK. Let's talk about cool things now.
Let's talk about science.
Exactly. Which he is not.
I picked a rabies paper because I knew Angela was going to be here, so you can enjoy rabies.
Let's go, rabies. I mean, that sounds weird, but yes, we like rabies. We like trying to protect animals and humans against rabies.
I don't know if we'll get to it, but we have a letter from someone who sent us an article. Where this lady was on holiday in Morocco and she was playing with a pet puppy and it scratched her and then she ended up dying of rabies.
Ooh.
What?
She didn't know she would be at risk. The little puppies just kind of scratched her. She said, she scratched my hand, or her daughter or something said, and then 2 weeks she started having all these symptoms. Of course, if she had been vaccinated immediately, she would have been fine. But I guess the moral is any contact with wildlife, you better get vaccinated.
Well, especially in areas where rabies is not controlled. So like in Northern Africa, dogs are not vaccinated routinely. Well, because there's a lot of stray dogs, they won't be vaccinated routinely. And there is a lot of rabies as opposed to in North America where we vaccinate all of our animals between like every 2 and 3 years. And it's something more controlled and we know what species are reservoirs. But if they're even dogs are reservoirs, then you definitely have to be careful.
Yep. Especially if they break your skin, right? She should have gotten a rabies vaccine, but she's dead. It's fortunate.
And
anyway.
actually, interestingly about this paper, just like so people are aware, over 60,000 people die of rabies every year still. Right. Humans. apart from animals, like humans die, 60,000, which is not an insignificant number. I mean, even one life is important, but that's pretty significant for something that we consider to be more or less controlled. But
Yep.
as your example, it's not.
Most of those are in Africa and Asia. There's a lot of stray dogs walking around. And so it's interesting. The dogs are the biggest source of rabies, not other animals. I mean, other animals can transmit it, right? Like bats and raccoons, but it's the dogs. Anyway, this is a self-hosted microbe paper, deep mutational scanning. I just like deep. Deep mutational scanning of rabies glycoprotein defines mutational constraint and antibody escape mutations. I would say they're antibody escape amino acid changes.
Yeah.
But throughout this paper, they're like rubbing it in TWiV's face almost. They use mutation all the time. The authors are Arjun Aditam, Kaylin Radford, Caleb Carr, Naveen Jasti, Neil King, and Jesse Bloom. Mostly from Seattle, Washington. All from Seattle, Washington. The Fred Hutch and the University of Washington. And this is about rabies, a Lissa virus, which is fatal. You get infected, you're going to die unless you get vaccinated. And as Angela said, 60,000 human deaths. They're all zoonotic because people don't transmit it to each other.
So I want to do a little for the folks out there, a little rabies background with Angela's help. So negative strand enveloped virus, right? It seems to be present in many different wildlife species, right? Bats, dogs, skunks, wolves, etc. Is there, in your mind, Angela, is there a dominant reservoir host, or is this, because this is unusual? It's all over the place.
Rabies in the wild. I mean, right now, dogs, I should say carnivores in general, like domesticated carnivores, but mostly dogs, are definitely the largest reservoir. But then in places like Africa, it's actually also dogs, but bats can carry rabies. Other intermediate hosts that are normally also carnivorous carry rabies, but other species, most mammalian species, or a lot of them can be infected by rabies. But the reservoir right now and danger to humans is definitely dogs.
Okay.
In most
so it's
contact cases, it's mostly dog contact.
So, and just to fill in a little background, it's neurotropic, transmitted.
Sorry.
Go ahead.
No, go ahead, Rich. Go ahead.
Were you going to say it replicates in non-neural tissue to start with, and then winds up in the nerves?
It's centripetally makes it up your nerves all the way to the nervous system,
right.
Exactly, and then
so transmitted primarily by the bites of animals that's in their saliva, okay, and then transmitted up the nerves to your brain, and that takes a while. If it gets to the brain, that's when you start showing symptoms, and if you've started showing symptoms, it's too late. You're dead. Okay, except in very rare circumstances, there are a couple of novel treatments that have been experimented with that have occasionally been successful.
And when he says a couple, he means like 2 people in the world.
yeah.
And it's that transit time that makes it possible to do post-infection prophylaxis with a vaccine. Vaccines are ordinarily, or it's therapy, actually. Most vaccines are prophylactic. You get a vaccine in anticipation that someday you may get infected. But in this particular case, the infection takes so long because, and as I understand it, Angela, it also depends on how far the bite is from your brain. How
literally,
long
if it was on your neck, or if it was somewhere in your anterior limb, like somewhere higher up in your body, it will be faster than if it's in your leg, for example. Right. That's actually important because it has to, especially imagine me, I'm like 5'10". If it's on my ankle, I have more time. The window is larger.
You're saying you're tall?
Yes, tall, exactly.
She has more time than I do, is what she said.
More time. My nerves are longer.
How tall are you, Brian?
5'2".
Oh, yeah. You're a shorty. I'm 5'7". What about you, Rich?
6 feet.
Wow, what a gradient we have here. I'm sorry, Brianne. I didn't mean to call you a shorty.
It's okay.
You're What do they say? Height challenge.
Vertically challenged.
Nah, it's fine.
So at any
But
rate,
the-
because it takes a long time, you can actually administer a vaccine and passive vaccination as well. That is immunoglobulin or anti-rabies antibodies after you've been bitten. And that is effective in getting rid of the virus before it has a chance to reach your brain and you have symptoms.
As far as I know, the initial, when people are bitten, you get immunoglobulin injected, like anti-rabies immunoglobulin injected directly at the wound site, actually. Ideally, the same day that you're bitten, to have, like you said, that passive immunity at the site of injection, at the site of the wound. But you can then also get the vaccination, which is normally an inactivated virus, inactivated rabies, that is given up to 7 days post-viral, like post-bite. Normally it's within 7 days. There is like a window. And then you get another one. I believe it's 14 days after. There's 2 doses, I think. And then there's also PrEP, so preventative exposure prophylaxis, which if you're working with animals in the wild, like if you're doing field work with bats, let's say, or I don't know, somewhere that has dogs with like rabid dogs, then you get also 2 vaccination. I think it's 2 doses as a human. And the vaccine is actually pretty intense. It's not like a slight, you don't really feel it or anything. People normally become quite febrile and get pretty sick, but it's better than dying of rabies, obviously. And for animals, your dog will normally get vaccinated between 3 and 4 months, their first dose, and then like yearly or depending where you live, every 2 years for the rest of their life.
And
Please
that
vaccinate
vaccine
your
quite
dogs.
commonly is poxvirus recombinant. Canary pox expressing rabies glycoprotein. The important antigen here, as we will discuss, is the glycoprotein, which is equivalent in a way to the SARS-CoV-2 spike protein that people are familiar with. Sits on the surface, is important for attachment of the virus, and is also the major site for antibodies to neutralize virus.
Your cat also should be vaccinated in case you're worried about them.
Right.
True. Although they don't carry, it's interesting that they're not reservoirs like dogs are. I don't know what it is specifically about cats. Like they can get rabies, obviously, but it's not anywhere near as prevalent in felines
Cats
as
are
it is
just
in
more dignified.
canines. Or they just don't really get close to people as much or other animals. Doing their own thing.
So the prophylaxis is 99% effective. So you wonder why there's 60,000 human deaths. Well, they just can't get it. And so we need cheaper and more widely available vaccines and antibodies. That's partly what this paper is about. So the main target of these vaccines is the glycoprotein embedded in the viral envelope. The glycoprotein allows the virus to bind to cells, and it also leads to a pH-dependent conformational change and eventually fusion. So this is one of those spike glycoproteins that has 2 conformations. There's a pre-fusion neutral pH conformation, and then the post-fusion low pH post-fusion conformation, which drives fusion itself. And they note that this is a problem for vaccine design because the most potent neutralizing antibodies target the pre-fusion conformation, but when you vaccinate, mostly you get post-fusion antibodies. So if that rings a bell, if you could somehow stabilize it in the pre-fusion conformation, you'd get the right epitopes exposed. This is basically the adding the prolines to the spike, SARS-CoV-2 spike glycoprotein.
Yeah, and I had to educate myself as I was reading because they talk about class 1, class 2, and class 3 fusion proteins from viruses, and I can never keep them straight. And so I didn't know if this was SARS-CoV-2-like or something else. This is a class 3, and I had to look it up and discover that SARS-CoV-2 spike protein is a class 1, and my sense is that those things are similar enough to one another so that the proline substitution trick that was used on SARS-CoV-2 can be used on others as well to freeze it in the pre-fusion conformation. This is a class 3. It does the same sort of tricks, but structurally and otherwise it is somewhat different. And from what I understand reading this paper, they haven't totally figured out how to stabilize this. There are some things they can do that stabilize it some, but they don't feel like they got a total lock on it. And part of what this paper does is help out with that.
All right, for 400 points, this virus has a class 2 fusion protein.
No.
No, I'm talking about what you have to answer it. This is Jeopardy.
Oh, Jeopardy. That's
Like-
another
I
one.
definitely do
Free
not
answer,
A
Jeopardy.
know.
flavy?
Flavivirus, yes. Would a flavy work, or do you need to say like dengue virus in Jeopardy? I don't know.
Yeah, I think they'd probably take flavy.
What is a flavivirus?
What is it?
Yeah, so we played Jeopardy at TWiV 1000, remember?
Mhm.
With technical challenges, but it still worked. It was fun. Anyway, so that's one of the things we're going to look at here, conformational changes. Also, there's diversity in G, 10% sequence divergence among naturally occurring rabies virus strains. So that makes monoclonal development a little difficult. Interestingly, here in the U.S., we use serum from people who have been immunized with rabies vaccine. But in India, there's a monoclonal that's used, actually, and other monoclonals are in development, which makes sense, right? Because then you don't have to use people.
You don't have to use
Yeah.
hyper-immunized people and taking
Yes.
their serum. Yeah.
Yes. So in this paper, they use deep mutational scanning, which is a thing. Deep means you put a lot of changes. So how many amino acids are there, folks? 20 amino acids. You want to put all 20 at each position of the glycoprotein. That's deep, folks.
And that's like
Wild.
431 positions, 20 amino acids. It's like 8,700 some odd
Yeah.
different
And
mutations.
They
Yeah,
get close to it.
Yeah, they say they did 8,227.
That's
So
pretty close.
what this is not done with rabies virus because it's a little dangerous to work with. So what they do is use lentiviruses, HIV vectors, where they include the gene for the spike protein of rabies virus. So now these lentiviruses are going to display the spike, and they can look at infection mediated by that spike. So they essentially make this lentivirus, and they substitute every amino acid with the 20 different amino acids, and they basically get 8,227 different amino acid changes, which they call mutations. But I won't say it again. And they also threw in a couple of stop codons at 20 sites as controls. Those should be dead for entry, right? And then they also, so they end up with 80,000 variants of G, and they barcode each one, which means they put a sequence that's different in each one so that they could always identify what the amino acid change is by the barcode. So, a barcode is the stuff on your groceries or anything else. I don't have a barcoded thing here, but you can scan it at the supermarket. It tells you the price, right? There you go. There's a barcode. These are not things that you scan with a QR reader. This is a short nucleotide sequence, which is specific for each change, and that they call it a barcode, which is cute, right? It makes a lot of sense. So they have 80,000 barcoded variants, which covers over 99% of the amino acids. And
So
most of these,
These are all, so they make libraries, right? So you
Yep.
have a population of viruses, 80,000 of them, each with a different barcode that have
Right.
been mutagenized in a fashion so that each barcode is associated with some mutation or other. And it's not perfect. They try and optimize it so that there's one mutation per virus, but a few of them have two, some of them don't have any. But as a population, all of the single amino acid substitutions in this thing, or nearly all of them, will have been covered. And since they're all barcoded, when you then use them to do an experiment and some virus comes through that experiment, you can identify what it is from its barcode,
right.
and identify which mutation is behaving in this particular way. Very clever, and I don't understand it all. Some of the technology is surreal.
I also was reading it and instantly Googling almost every
Yeah.
third sentence. I was like, what
Yeah.
did they do here? What is this?
Yeah.
I was like,
Yeah.
I'll need Rich, Vincent, and Brienne's help to understand what this means. The take-home message is very interesting, but yeah, they did some very...
The details of the technology are tough. It would take me another couple of days to sort it out.
So the cool thing is this barcode. You may wonder, how do they know the barcode? Well, if they when you infect cells with these lentiviruses, because they are retroviruses, the RNA is copied to DNA, and then you can just extract that and do PCR very quickly for the barcode.
Oh yeah,
So
that's the other important thing about this is that it sounds a little scary that you've got a rabies virus glycoprotein in an HIV vector or something like that.
I was going to say that too. We should explain what that means.
But really what this is a particle, a virus-like particle with the rabies virus glycoprotein on its surface. And the whole lentivirus thing is just a technology for getting that particle. And that particle, since it has rabies virus glycoprotein on its surface, can, if everything is working right, attach to cells and enter. And then because it's retrovirus stuff, it can be reverse transcribed into a covalently closed circular DNA, which can be detected by doing appropriate nucleic acid stuff, but it doesn't get any further than that. Okay.
Right.
So it's dead.
There's no HIV, infectious HIV or
it's
anything
dead.
like that.
So basically, the other thing that's really nice about this is it also isolates the part of rabies infection that is that entry step.
Yeah.
And so it just uses this G protein, so we can just see what's going on with entry mediated by the G protein. And that specific thing is what we want antibodies to block. And so if entry happens, then the cell acquires this new piece of DNA that has been integrated in the way that Rich describes. And you can say, oh, that virus entered. Let me see which one it was, by seeing which barcode of DNA got into the cell. If the virus didn't enter, that means the antibodies blocked it and its barcode doesn't come up. So you sort of can see the difference between which ones were able to enter and which ones were not.
And I guess you sequence the library, right? So that gives you, that associates a unique barcode with every G protein sequence. And whatever mutations happen to be there. So from then on, you have you can identify what G protein you're dealing with just by knowing the barcode. And the barcode is a random sequence of 16 nucleotides. Okay, which if you figure that out, that's plenty of different combinations.
So the first experiment, you take this library with all of these changes, you infect cells, and you just extract the DNA and then sequence it, and you say how many barcodes made it inside.
Right.
Because some of the changes are going to mess up entries, they're going to mess up binding, they're going to mess up fusion, and they'll never make it inside of the cell. They'll never make it to DNA. So that gives you a whole lot of data originally. You could do one experiment in theory and get a lot of data. Now, of course, you want to do the antibodies, you have to do a separate experiment for each antibody, as you'll see. But in theory, if it worked, you could do one experiment. But as you know, never works the first time.
Or if it does, it's a fluke, and then it never works again.
That's right. That's right. So that's how this works. It's all about sequencing the barcodes. And then they actually say, okay, we've identified this amino acid is important. They could make a specific virus with that amino acid change and test it just to validate. And they do that often as well. So what they do is they make a cell entry score, right? And greater than 0 means whatever change has improved it, entry, and less than 0, the entry is impaired. And so the stop codon insertions, of course, are 0 because they don't get in at all, right? So what do we find? Okay, most changes have either a neutral to slightly deleterious effect or a highly deleterious effect. So not much of a change or a lot of change. And the effects vary across the sites and even at different changes at the same site.
And no positive effects.
No positive effect, yeah. now they have the structure of the G protein, which is nice because then they can put all of these changes in the context of the structure. And so if you look across the whole sequence of the G, you get different results in different parts because there are different functional domains of the protein. There are some highly constrained sites where you can't touch it. Nope, can't touch it at all, can't change it. Or there are tolerant sites where you can put lots of changes in. These domains are called three central domains: the plextrin homology domain and the fusion domain. Plextrin is just another protein that happens to have homology with it in the fusion domain. And then there's some, so for the constraints, structural constraints where a protein has to fold in a way, and the amino acid is important for that. Amino acids that are buried inside the structure. Remember, proteins have a three-dimensional structure with amino acids on the outside and the inside. The ones on the inside tend to be hydrophobic. So if you stick a non-hydrophobic amino acid in there, it's not good. It's going to disrupt the structure. The fusion loops that are important for fusion of the viral and cell membranes, those are also highly constrained. And then there are sites that are important for this conformational change. Remember, we're going from a pre-fusion to a post-fusion conformation upon low pH, and they identify amino acids that are important for that. In particular, histidines. There are a cluster of histidines that, and this happens in other proteins as well, they're pH sensors, right? And they're important for that conformational change. So if you change those histidines, you're dead in the water because you can't sense the pH change. And then, of course, they have sites where the change is not good because of the conformational change that's needed. So normally the G protein is in an equilibrium with pre and post fusion conformations rapidly varying, but they find some amino acid changes that stabilize the extended conformation, the post fusion conformation, and they say this is a pre-fusion. They find some changes that stabilize the G protein in a pre-fusion conformation, akin to the adding two prolines, right, to the SARS-CoV-2 glycoprotein. They say these are candidates for stabilizing mutations that could shift the conformation towards the pre-fusion state. Again, that's where that would be for a vaccine where you have the proper epitopes exposed.
I want to talk about proline for a minute.
Sure.
So
By the way, Rich, when we were in Austin and we interviewed Jason McClellan, which wasn't too long ago, I think it was last spring, he talked all about this sort of thing. Right? Yeah,
great stuff. So one of the interesting things about this analysis is that there are sites in the Remember, we said on the top of this that they know some about how to stabilize the protein. They'd like to know more so that they can really lock it in a pre-fusion complex. And the trick that we've talked about repeatedly is proline. And the deal is proline's a special amino acid. Most amino acids have it's hard to describe without going into the structures, so I'll try and just give you the overview. Have a lot of flexibility, in particular rotation around various bonds in the chain, so that normal amino acids in the linear chain can rotate and flex around any given amino acid, but proline is special because it's the amino group is hooked to the alpha carbon, and that restricts that rotation, so it's much less flexible. And you find it in turns in proteins where you need some, where you benefit from that sort of rigidity and the configuration that comes with it. And if you introduce it into sites, it can, because it's less flexible, introduce some rigidity. And one of the interesting things about this analysis is that, remember we said we changed all 20 amino acids to each site. There are some sites that are sensitive to that, where entry is compromised, but are compromised specifically by prolines. Okay, and the other amino acids you can change, it doesn't make any difference. So that is a clue that those sites are sensitive to this flexibility issue and might be, maybe the problem is that if you stick a proline there, you're locking it in the pre-fusion complex, so it can't undergo the conformational change that's necessary.
So if people wanted to know a little bit more about what Rich was saying, I'm going to super oversimplify, and I apologize in advance to the biochemists who will be appalled. But in some ways,
else will thank you.
In some ways, you can think of an amino acid as sort of having 4 different parts. Almost think of it like a plus sign. There's the left and the right side and the up and the down. Basically, these are 4 different things coming off of one carbon. And in most amino acids, there's sort of 4 different chemical species on each of those 4 parts of the plus sign. In proline, the left one and the bottom one are attached together.
Right.
Or the left one and the top one or something, but depends on what direction you have is up. But in any case, you've got a little connection between the 2 arms that is not present in any of the other amino acids.
That makes a kink in the protein.
Exactly.
Right?
You can actually, if you're a crazy enough TWiV listener, you can go look this stuff up and look at the structures and look at what a polypeptide is and look at what an amino acid is and look at how different proline really is.
So, in fact, people previously have put a proline at position 270 of the rabies G protein to stabilize it in the pre-fusion conformation. And in this study, they find that, in fact, their deep mutational scanning, if it puts a proline at that position, it's dead for cell entry. And that makes sense because if you lock it in the pre-fusion conformation, it can't fuse to get into cells. But, that's for a vaccine where we can lock it, and that's good for the vaccine. But they're interested in, are there other regions that we could use to lock it into a pre-fusion conformation? So they look through their data to see where prolines have been problematic for cell entry, and they find a number of particular site, and they validate those by making viruses to show that they're additional candidates for kind of putting a proline in to stabilize. So you don't have to put it at just one place. You got a couple of different candidates where you could put it. So that's cool for what Jason McClellan calls vaccine engineering, right? Protein engineering to make vaccines. Okay, so that's the overview of the protein you get from this deep scanning. Now they go into the antibodies. How do these amino acid changes affect neutralization by antibodies? And again, if you, as Brian said before, if you add an antibody to the lentivirus, and then in fact, if the antibody is blocking infection, you will not see the barcodes get inside of the cell. And if the amino acid change makes you resistant to the antibody, then the barcodes will get in the cell, right? So there's an antibody called 17C7. This is used as a post-exposure prophylactic. I believe, it's deployed clinically in India as of 2017. So they use that there. So that has had its structure solved with G. So they look at the effect of their amino acid changes on neutralization, and they see many of these are. So they find antibody escape mutations to that particular monoclonal antibody. And since they know the structure, they know exactly where the antibody is binding to the G protein. And it turns out that and remember, they're doing their deep mutational scanning and then doing neutralization assays. You only have to do it once, right? You just take your whole library, you mix it with the antibody, then you infect cells and you sequence, and you get all the data. And they find that a lot of escape mutations are tolerated because they can see the virus getting the barcode getting into cells. And so they identify basically escape changes at the interface between G and the antibody, as you might expect, right? They're all where the antibody is hitting the G glycoprotein. One of them is not in the contact area, I338T, but creates a glycosylation site, which interferes with the antibody binding. Isn't that lovely? Just exactly as you would predict. That happens with other glycoproteins as well. And then they go to the other antibody. So that was one antibody. And they do the same experiment with 8 monoclonal antibodies that have been made to target G. They bind to different regions of the glycoprotein. The 17C7, as I said, was deployed in India, and the others are in phase 3 trials. And they all neutralize the pseudovirus, the unmutated pseudovirus with different potencies. And so again, you take the library of mutations, you mix it with each antibody separately, and then you infect cells and you sequence and look for what barcodes get in. In other words, which amino acid changes get into the cell in G. And again, each antibody, they see escape to each antibody by a small number of changes that are in the prefusion structure of the G glycoprotein near the antibody binding interface. And interestingly, they don't see changes at every amino acid in the contact site. Some of them are lethal, and you never see any escape because those changes are needed structurally, for example. But other sites tolerate a lot of amino acid changes, and you can get escape at those as well. And even if you have an antibody that two different antibodies that target the same site on G, they can have different profiles in the escape amino acid changes that lead to resistance, which
right,
is
because the binding site is not exactly identical
on the two antibodies, right?
Yeah.
Right, different sequences. Okay, so this is interesting because they have antibodies that have broad neutralization potential. So remember, there are lots of different isolates of rabies virus. So some antibodies can neutralize a lot of them, and some of them have a narrow spectrum neutralization. And they find that the broad spectrum antibodies are, as you might expect, escaped by changes at fewer sites than the broadly spectrum neutralizing antibodies, right? So that's kind of interesting there. So let me say it again. Unlike the narrow spectrum antibodies, the broad spectrums are escaped by changes at fewer sites. Okay, and that's why they are broad spectrum, because it's harder to get escape for them. And finally, they say, well, what's out there in nature? We're using a Pasteur strain of rabies virus. What about what's out there in nature? And so there's a lot of sequences out there, right? There are 7,122 available G sequences in the next strain database. And they can take those sequences and ask, based on their data, which would be resistant to antibodies, right? It's not even a wet experiment. It's just a computational experiment based on their data. And it's interesting that most of the isolates lack escape changes from 17C7, which is the one that's being used in India. I mean, there are a few, but most of them, there's no escape. And most of the escape G proteins to 17C7 are either from bats or wild animals, not dogs, right? Dogs are the most common source of human infection. So that tells us we're not going to see naturally occurring resistance to 17C7 most likely, right? So that's the value of this study. You could design a monoclonal that targets a site that doesn't vary very much based on their mutational scanning, right?
I think it would also be interesting to do it in a cell line that is maybe more relevant to where rabies would naturally replicate, either in like a skin cell line or even neuronal cultures. I know that some people have done like rabies experiments in neuronal cell lines, but this is like 293 cells or what, kidney cells, which
they're kidney cells.
Yeah, kidney cells. They're like I'm just saying it would be interesting to see if the same would hold true in a different cell line, because kidney cells will never necessarily be infected with rabies virus in like a physiological circumstance. But
I think you could take a subset of them, or you could just do the whole library and see if you get the same result. It's not really hard to do. All you do is infect and sequence, right?
That's what I mean. They already have, like, the hard part's already done. You could
to see if there's any difference, so how the protein is presented on the surface of a different cell type.
Yeah, I mean, I guess the issue is since they're using pseudovirus, does it really matter about the cell type?
Yeah.
So we were talking in the pre-show, wasn't it, about classic papers?
Yeah.
We didn't do that publicly. We some time ago in TWiV, we did a bunch of classic papers. We didn't ever do Pasteur's rabies vaccine, did we?
No,
That'd be
I don't
cool. We should do that.
Because that would be really cool. I'm thinking about this because of the Pasteur strain. And for, once again, the audience out there, Pasteur was one of the, following Jenner, of course, one of the original vaccinologists, immunologists. And one of the things that he did, capitalizing on what he'd learned from Jenner, is to try and figure out how to make a rabies vaccine. And my guess is that this particular Pasteur strain of rabies virus is somehow connected to those original efforts. That would be an interesting story all by itself. But the story of Pasteur's development of the rabies vaccine, I won't go into it, is absolutely fascinating. I would be willing, if I can find some time, to assemble those papers into a package, and we could do a classic story of rabies. Angela,
That'd
you
be
would
cool.
have to be on that one.
I would definitely.
What an amazing
That's
story.
like 1885. I
Yeah.
just checked because I wasn't sure when. And there's like pictures of his like physically writing it down. That paper is.
Yeah, it's
Would
really
be
amazing.
wild.
Yeah, no, it's very cool. And if people are also interested in this, I once had to watch the award-winning movie, that is a story of Pasteur's life. I believe it is called The Life and Times of Louis Pasteur. So I would recommend watching that movie because you
Does
learn
that
lots of
dramatize
Pasteur
the
fun
rabies
facts.
vaccine
It
thing?
dramatizes all sorts of
Okay.
key experiments that Pasteur did.
What an amazing dude.
Yeah.
Yeah, there's something. So that's the paper. We have a map of the glycoprotein, what sites can tolerate change, what sites can't. But more importantly, these 8 antibodies, how you can block their attachment. And what I think is really cool is that in the wild, you can find resistance to all 8 monoclonals, right? Although how much, some are more common than others, as we talked about. And you could use that to design a monoclonal, right? To say we're going to target this place, which has is not likely to be changed to give you resistance.
There's an interesting thing going through my mind as I'm reading this paper too, and that is the concept of fitness in this kind of analysis, because there's 2 issues going on here when you're doing these assays. If you have an amino acid change that makes it so that the antibody can't bind, but we sort of mentioned this, but that same amino acid change makes it so that the glycoprotein doesn't work anymore, okay? Then that doesn't score as something that is an escape mutant, okay? And this happens in nature. You can't just do any old mutation and that is in an antibody binding site and compromise the antibody binding, because some of those mutations will make it so the virus doesn't work. In nature, that's what we would call a fitness cost for a mutation. Yeah. Same goes for drug resistance. Finding antiviral drugs, the trick is to find drugs where almost any resistance mutation that you can think of will create a fitness cost for the virus so that you don't get a lot of resistance to the drug.
Or you might say a situation where there's a structural constraint.
Yeah. Yep.
So here we only looked at entry, but
yeah, exactly.
It could be that if you put these in virus, you would have different outcomes, right?
Mm-hmm.
So you could imagine that virus could get in, but it would have some other defect in the subsequent cycles.
You could have a problem where the fitness cost is because once you initiate the virus cycle, the protein doesn't fold correctly in the cell or something like that and can't be assembled into the virion, okay? Maybe if you do it artificially, right, you can make a virus that works. But
Yeah. Yeah, it's all about fitness in the end, folks. If you just think about that, fitness, the ability of the virus to reproduce sufficiently to do all the things a virus has to do, including transmit to a new host. Okay, that is paper one. This and then
that was
the second one.
That was a snippet, right?
That was a snippet. It was a half hour, right?
Yeah.
Well, there's a snippet in name.
Yeah.
I think
There's no
this-
such thing as a real snippet on TWiV, I don't think.
second paper is in the ISME journal. What does ISME stand for? Anybody know?
Oh, I looked it up.
International Society.
Of microbial ecology.
Microbial
probably.
ecology. Yeah, I think that's it.
ISME journal. Incomplete tricarboxylic acid cycle and proton gradient in Pandoravirus massiliensis. Is it still a virus? And yes, it is. Okay, we're going to
So
end
wait,
that.
Can we
Yes,
start?
It totally
Can we
is.
start for all of the listeners with, I mean, you, Vincent, Rich, Brienne, whoever wants to take this. What, right, as of right now, is the definition for the public, like for the lay public, let's say, how would you define a virus? What does it have to do or not do to be a virus? Is it alive? Is it not alive? We don't have to say that, but I mean, what is the definition of a
The
virus?
definition of a virus that I give in my course.
Mhm.
It's an infectious obligate intracellular parasite comprising DNA or RNA with a protein shell and sometimes a membrane. But the important parts there are obligate intracellular parasite. It needs to get inside of a cell in order to reproduce. But there are other nuances that you can think about.
Brienne
Yeah.
has a nuance. I see it.
Yeah,
Let's
See,
yeah.
Brienne.
So that is not exactly the definition I use in my classes. So I talk about an obligate intracellular parasite of the cell's protein synthesis machinery, particularly an obligate intracellular parasite of the ribosome, because every virus needs to use its host cell's ribosome to do translation. There are obligate intracellular parasites with other kinds of microbes as well, but they are using some other part of the host cell. So if I think about something like chlamydia or Wolbachia or Rickettsia, those are also obligate intracellular parasites. Nobody ever says, oh, they're not alive or, oh, they're a virus. And the real difference is they have their own ribosomes, while viruses need to parasitize the ribosome. And I like Vincent's part, having DNA or RNA, not having DNA and RNA. And the other thing that comes up in a couple of places in one of the micro textbooks I use is disassembly upon replication. And so in order to replicate, you actually have to completely come apart as sort of key pieces of a virus. And so I've kind of always thought about the definition in terms of how do you exclude the other obligate intracellular parasites. And so that's where how I sort of phrase the definition, is really kind of translation focused or disassembly focused.
These Rickettsia and chlamydia, they need something else from the bacterium.
Exactly.
Right?
Yeah.
But they
have
can.
Yeah. They can divide, and they can do translation, but they need something else from the bacteria, not the ribosome.
I mean, most viruses. I mean, all viruses need ribosomes, as you say, but most viruses need a lot of other stuff too.
True.
You know, only the biggest ones have some metabolism genes and some lipid synthesis genes and so forth, but none of them are complete, so they could never exist on their own. They still have to get into a cell beyond the ribosome requirement.
Yeah, but like chlamydia can't exist on its own.
So chlamydia will not divide outside of a cell, right? Any of those others, can they divide outside? Rickettsia cannot, no?
Mm-mm.
So one of the nuances that often creeps into descriptions of viruses is that they do not have their own metabolism, energy metabolism, okay, that's becomes important for this paper.
I mean, even from this paper, it's not clear that this virus can make its own energy.
No,
we'll
get into that.
It has some signals, right?
Yeah.
But anyway, this is from a group at the in Marseille, okay, at the university and a few other institutes. And first, the authors are Aherfi, Belhawari, Pinot, Baudin, Decloumont, Abraham, Colson, Levasseur, Lamb, Chabrier, Raoul, and La Scola is the last author. This came out in 2021, and I remember when it came out, and I said, I'm not sure I want to do this. But last week we had a textbook meeting, and we discussed it, and suddenly it became interesting to me. I'm not sure why, but I mean, I like, you'll see, it's got some metabolism, and I think metabolism is cool, despite having forgotten it all from high school years ago. In the context of viruses, it's very cool.
I never learned it. I never took a good metabolism course. I taught in a biochemistry department for 12 years, and I never took a real biochemistry course.
So
I had to kind of learn it,
that's
on
fine.
the
When
job.
you can learn it later. It's a lot easier. My high school biology teacher, she taught us the Krebs cycle, and she said at each step there's a little puff of energy. And I'm like, what the hell is this puff? She drew a cloud on the board. If she had said ATP, I would have got it. But she never mentioned ATP. Puff, a little puff of energy. No, sorry. Anyway, so this is motivated by the study of giant viruses, right, with genomes bigger than we expect, right? Mimi virus, the first giant virus in 2003. And, the authors say they challenge the definition of viruses, but no, they actually don't, because they just have a lot of genes and they're really big. It doesn't challenge the definition of viruses. They still need ribosomes. They still need to get into a cell. But this paper deals with Pandora viruses. The first one, Pandora virus salinus, the first Pandora. So it's called Pandora virus because when they sequence the genome, it's really big. It's 2.5 million base pairs. Million. I mean, that's bigger than some free-living organisms, okay? And a 1 micron diameter viral particles. When they sequence it, like 90% of the genes are dark matter. 80% of the gene open reading frames are orphans. It's a good name. I really like that.
Orphan with an F.
ORF, open reading frame, because we don't know what the protein. There's nothing like it in any database. So they said this is really unusual. Then over the years, more of these giant viruses have been isolated. Some of them encode proteins involved in translation, like Tupan virus encodes almost a complete translational apparatus, except it's missing one thing, the ribosome, which is really important. Got all the tRNAs and a lot of other things, but there's no ribosome. So it still depends on the host cell. And then over the years, they find other genes encoded in these genomes, like cytochrome P450 monooxygenases, other genes involved in metabolism and fermentation, sphingolipid biosynthesis. I used to say in my course, no virus encodes anything for membrane biosynthesis. Boop, that's out, because they found genes in some of these giant viruses. And the idea is generally that the viruses make these products maybe to tweak the host cell into doing things more in their favor.
Yeah, it's not necessarily that the virus is going to do any of these processes. It's going to try to push the cell to do processes in a way that might be good for the virus.
And they write here, this just bugs me, as giant viruses have challenged nearly all criteria defining a virus. No, they haven't. I mean, they're interesting and they have unusual things, but they don't, as Brienne just said. Anyway, they said that one of the last things we don't know about is energy. Can viruses produce energy? And so they look at another Pandora virus massiliensis, which they isolated not too long before this paper was published. And as you'll see in this paper, they seem to have some properties that are associated with energy generation. And that's why I think it's interesting. And it's not because it makes it a cell. It's just that it makes you think about the origins of viruses, right? If this virus has these components, so where did it come from? Where is it going?
Do you think that it could be some sort of, I mean, we have to go more into it, but some sort of like virus cell continuum or an intermediate phase of life? It's kind of like that paper you did on TWiV with Nels. It's like, what are you?
I don't know. It could be that this came from a cell and this is now degenerated to the point where it doesn't need to make energy because it's going into a cell. So it's lost almost everything, but not quite. Right?
Yeah.
Maybe in a million years, it wouldn't have any of these things.
Yeah.
Exactly.
I guess I've kind of, you're talking about it as a continuum. And I like that because I think I'm very frustrated sometimes when people are like, it's a dichotomy, yes or no. And I think that viruses like this, viroids, there are prions, like there are a lot of different things that you could say are sort of on this continuum, the gray area between yes and no. And so I think this is just another example of something in the gray area.
I will promote my Principles of Virology chapter again, because at one point in trying to sort out for myself and describe to others what taxonomy was about, I said, in the end, biology is a continuum, but scientists are burdened with the responsibility for drawing boundaries within that continuum so that we can talk about it.
Yeah.
All right. But I think you should keep that continuum concept in mind all the time. The boundaries are artificial. Nature doesn't know. Nature doesn't know about those boundaries.
I think it's been even more clear now within the last 10 or 15 years with single cell sequencing, where you can see so much more in subsets of cells, if we're talking about cells, not viruses even, which I deal more with, that we thought were discrete populations. And we see that they're actually like a continuum, and that like something that you thought was called, let's say, a T cell, like a Th2 cell or a Th1 cell, depending on the organ that it's in, like transcriptionally can be very distinct. So like everything is kind of, single cell I think has highlighted that if you look at cells specifically, like we can look at each cell's transcriptome, we see those differences. Obviously with viruses and sequencing, we've also seen that. And that's been very eye-opening, where before, even just 20 years ago, people would challenge that. But now I think it's a lot easier to see with that type of technology.
Yep. All right, so the first thing they look at is whether these virus particles, these big Pandora virus particles, have a membrane potential, right? So what's that? So in the mitochondria, which make, there are batteries, right? They make energy, a lot of energy. So you can make some energy by glycolysis. You eat food, it gets degraded to glucose, that gets taken into cell and metabolized all the way to pyruvate. That makes about 20% of your energy. But the 80% comes from the mitochondria. And what happens there is that the food which you eat eventually is broken down. Pyruvate is the end result of the degradation of glucose, and that goes into the mitochondria and drives energy production. And the way it does it is that the oxidation creates a gradient of protons across the mitochondrial membrane. So there's more protons on one side or the other, an electrochemical gradient, which is what they're looking for. So that electrochemical gradient is then used by amazing ATP synthase enzymes to make ATP, most of the ATP that we need. And by the way, you need 2 molecules of ATP to add 1 amino acid to a protein. So we need a lot of ATP, and that's just protein synthesis, right? So this proton gradient is the key to life.
Yeah. If I was going to think about it, and you're going to hate this one, Vincent, too. When we're trying to do metabolism in, say, the mitochondria or something like that, if we did a lot of energy release steps, if we did a lot of bond breaking in big steps, we might lose some of the energy. Some of it's going to get lost to heat. And so instead, we try to do it in little bitty steps that aren't even enough for a full ATP. They're like a poof or whatever you said.
A puff.
A puff. Yeah, they're a little bit of energy. Not enough to make a whole ATP, but enough to move some protons. And if you just move a whole bunch of protons and save that up, then you can get tons of ATP later. So you have to move a lot of protons, make this big charge gradient that you're going to use to make ATP later.
Right. So that's fine. I don't have a problem with that.
I just thought I'd bring back your puff of energy.
There is a nice article I found on proton gradients. It's in Nature Why are cells powered by proton gradients? And it is co-authored by Nick Lane. Well, it is authored by Nick Lane. Nobody else wrote it. Who's written a bunch of really good books for lay people on this subject, which I like. And he sort of gives a history of this proton gradient. And what I love is how, remember Ephraim Racker?
Yeah.
So he's the one who figured out how glucose is degraded in glycolysis to give a little energy. And he makes this point saying this is a chemical reaction because everything is accounted for, right? This chemical goes in and this chemical comes out. But where you get 80% of your energy is actually not a chemical reaction. It's this gradient, which he said is not stoichiometric. It's a gradient. So you get intermediate. He says you actually make 1.5 ATPs per reaction. And of course, there's no such thing as 1.5. And that's the whole point, is that we store these extra half ATPs as electrons. It's a really good article anyway. You should go read it. It's open access. But that's why he says the flow of protons, I'm quoting, through these turbines in the mitochondria, the ATP synthase, which is actually a freaking turbine. You can see a movie of it because they solved the structure of the whole bloody thing. And you can see this thing cranking, and it uses that gradient to make ATP. It says much the way that water through mechanized turbines generates electricity. So the electrons power this turbine, and it makes ATP.
Yeah.
And go ahead.
All of that. One thing that I think that will come up later, because I think both Rich and I sort of typed about this in the comments. The way you get that proton gradient and do all of the things that Vincent just mentioned is there is this thing called the electron transport chain.
Right.
That's really important that actually sets up the gradient.
Right.
And so that's a question I have about this paper. So basically at this point of the paper, they're showing there is a gradient, but they don't really show us where it came from or how they got there.
Right.
And so when I see that, I'm like, oh, there must be electron transport chain somewhere. And so, but they don't actually show that, and they only talk about it a teensy bit in a couple places of the text.
So let me do my 2 cents on this. And this is coming from the guy who never took a biochemistry course, right? So I can look at it in my simple-minded fashion. In this whole energy metabolism process in mitochondria, I see 3 fundamental processes. One is what we're going to talk about in this paper, which is the citric acid cycle, also known as the Krebs cycle, that Vincent has introduced, where you start with pyruvate and it gets metabolized through several intermediates and that kind of stuff. And the major product of that, 2 major sorts of products, one is called NADH and the other is called FADH, okay? These are generated from NAD plus and FAD plus by the addition of electrons. So they're molecules that essentially can be electron donors or electron carriers, all right? Mm-hmm. They themselves, once you've made those, as I understand it, inside the mitochondria, and that happens inside the mitochondrial matrix, you don't have a proton gradient. Second component, okay, is what Brienne mentioned, which is the electron transport chain, which is absolute magic. These are proteins in the mitochondrial membranes that can take electrons from these molecules, NADH and FADH, and through a series of enzymes, catalyze a process that pumps protons, positively charged ions, out of the mitochondria. Now you have a proton gradient. And then the third bit of magic is because you have a proton gradient, those suckers want to get back in because you got more outside than inside. And the way they get back in is through this thing Vincent described, which is the ATP synthase. They come in through that thing and drive its rotation, and that generates ATP. So 3 things: citric acid cycle, OXPHOS that gives you oxidative phosphorylation that creates the proton gradient, and then finally the ATP generation.
And so the TCA cycle is the source of the electrons, essentially, that's driving this.
That's right. But the way I see it, And somebody who knows more biochemistry than I do can contradict me if they want. I'd be enlightened. The citric acid cycle by itself doesn't generate a proton gradient. Okay?
Exactly.
No, that's right. That's just correct. It just makes available NAD, as you said, with proton NADPH.
Yeah, I think of the TCA cycle slash tricarboxylic acid slash citric acid slash Krebs cycle, whatever cycle we're calling it, loads up electron carriers with electrons. Mm-hmm. And then those electron carriers have to pass off the electrons and get recycled so they can get reloaded up.
Right.
And I love
this is all aerobic. This depends on oxygen. Yeah. Whereas the glycolysis that we talked about is anaerobic. Right. One of the end products is lactic acid. So if you're running or lifting weights and it hurts your muscles, it's because you're accumulating lactic acid. You don't have enough oxygen, so you're doing anaerobic glycolysis.
And who put this thing in the show notes about the real Krebs cycle?
I did.
It is true. It's super true.
So it's really great because we all learned this at some point or another. It's really complicated. So Vincent put in this thing. It's a graphic that's the real Krebs cycle, which is you learn the Krebs cycle, then there's an arrow going over to forget the Krebs cycle, and an arrow going back to learn the Krebs cycle. So I just relearned the Krebs cycle again in the past day or so.
See, this is why I love this, because it integrates so much stuff. And, it's amazing that it works. These proteins in the mitochondria, holy cow.
Holy cow,
it's just
indeed. Yeah.
So when your mitochondria stop working, you're screwed. And when they stop, that often induces programmed cell death to get rid of those cells because we don't want them messing up everything.
That's a good episode title. When your mitochondria stop working, you're screwed.
Anyway, so back to the paper. How did they show there's a protein gradient? Well, there are a couple of dyes that you can add, which they will stain mitochondria based on this electrochemical gradient. They are specific for mitochondria that have this gradient across them. So if you poison mitochondria with, say, cyanide, the gradient, or something else, the gradient goes away. So they stain virus-infected cells and mature virus particles. They're called MitoTracker and TMRM. And these are fluorescent dyes. You could look under the UV microscope, and they see that a fraction of infected cells and a fraction of particles stain with these dyes, which, would stain mitochondria. It's a typical way of seeing mitochondria in cells. And they have cowpox virus, and they don't stain. So cowpox virus doesn't have this electrochemical gradient. And they have a lot of controls that they do, but you can see nice pictures here. And they also use an antibody to a viral protein to show that we're actually looking at viral proteins. So the MitoTracker is red, and then the antiviral antibody is green. And you can see there are a couple of particles that are red and green, and a bunch that are just green.
There's only two.
There's only two in that field. But they say 20% of the total particles are labeled by both virus antibodies and MitoTracker, although you wouldn't see it from that picture, right? Yeah. Or maybe that's 2 out of...
No, it's about 2 out of 10.
10, yeah, that's 20%. Yeah.
So that's the evidence. They have more images than just this.
I'm sure.
Hopefully.
So then they have an inhibitor of this electrochemical gradient, CCCP. It's kind of appropriate, the old name for the Russian name for the Soviet Union, right? Mm-hmm. It destroys the electrochemical gradient, and it also decreases the staining with these dyes. So that's the main evidence that these virus particles, remember, have some kind of electrochemical gradient. Although, as Rich said, we don't know how it got there. They don't. Well, next they're going to look for citric acid cycle containing genes, which would be a hint. But they don't find any ATP synthase or any of those electron transport genes in the proteome of this virus.
Yeah, I found that kind of interesting, both in the abstract and here in this part of the paper. They just sort of say, "Oh yeah, we didn't find that," and don't really mention anymore. And I was like, "Wait, that was the kind of important part."
No, I mean this. I figured the discussion around this paper would actually be more interesting than the results.
Yeah. So
That's why I wanted to talk about it.
I throughout this whole thing, I've been trying to understand how MitoTracker works. And they say, because does that actually mean that there's a proton gradient? There is an electrochemical gradient. Is that the same always as a proton gradient? Or can we have other kinds of electrochemical gradients?
It's just membrane potential.
Right.
Right.
Okay.
So it doesn't have to be protons. Yeah.
I'm coming back to this proton thing, okay, and how it was established. And we haven't talked yet about the CCCP, right?
The CCCP, which is the depolarizing agent, right? It removes membrane potential.
Right. But that thing is described as an ionophore. So it's basically just essentially poking small holes in the membrane and destroying that potential. So whatever it is that is causing a membrane potential, the CCCP, whether it's protons or something else, could do the same thing.
Yeah.
Okay?
That's true.
So I'm not convinced that any of this says that we have a proton gradient. Okay? Doesn't mean that there does seem to be something that makes the MitoTracker get in. All right? That's it.
Agreed. So then they're like, why do we need a membrane potential at all? And they suggest it may be important for viral DNA release, and they cite some examples with other viruses where that's been suggested. For example, chlorella viruses have potassium channels in them, and if you inhibit those, it prevents release of the DNA into cells. And there are some other examples that they cite as well. In fact, the results are mostly them talking about other results and summarizing other papers. But the next experiment they do is they add some acetyl-CoA. So if I remember correctly, so pyruvate is the end result of glycolysis, but pyruvate can't get into the mitochondria. So it's converted to acetyl-CoA, right? And that can get into the mitochondria, right? So they add some acetyl-CoA to their virus preparations, and their MitoTracker TMRN fluorescence goes up in the presence of low concentrations of acetyl-CoA. Okay, so why is that interesting? Because if you're driving the Krebs cycle, which acetyl-CoA would do, you're feeding into the electron transport chain and you're making a gradient. So it's really indirect evidence that what we see is a proton gradient, Rich, Of course, when they put high concentrations of acetyl-CoA, the gradient dissipates. I'm not sure why that is. Does anybody understand that?
I would have to understand the Krebs cycle better to understand
You
that.
can't overload the TCA
I feel like
cycle,
it's something akin to product inhibition.
Maybe.
Maybe.
Anyway, they say this demonstrates that the membrane potential that we're seeing is modified by acetyl-CoA, which of course is a regulator of the TCA cycle. So then they say, okay, let's see, are there any of the genes? So the TCA cycle, of course, is a set of 8 reactions. You put in acetyl-CoA, and then it gets converted to citrate, isocitrate, alpha-ketoglutarate, etc., all around to oxaloacetate, which then goes back to citrate. And so, and at almost each step, you make a little bit of energy. And so the enzymes, there are 8 enzymes that do this, like citrate synthase, isocitrate dehydrogenase, fumarase, my favorite. I like that name for some reason. And so they look for the genes encoding these enzymes in the protease, and they find some genes with low homology. 8 gene products encoding citrate synthase, aconitase, isocitrate, isopropyl malate dehydrogenase, and alpha-ketoglutarate decarboxylase. Okay, so these are enzymes in the Krebs cycle. They find genes that encode proteins with low homology to these. They look in other Pandora viruses, and they don't find them. Okay, so they look for other proteins involved in energy metabolism. Nada. Okay, all they find are these 8 genes encoding putative TCA cycle members. And so they say it's really a partial TCA cycle. It's not enough to do the whole thing.
And the homologies are
pretty low.
What was our word? Sketchy?
Sketchy.
Questionable.
As I'm reading this, I want to sick Eugene Koonin on this. Okay?
Yes.
And have him do the appropriate homology searches and see if he agrees with the identification of these as potential Krebs cycle genes.
So they say here, as this virus is neither a bacteria nor a eukaryote, the role of a partial TCA cycle and a membrane potential is enigmatic. They found no evidence that the particles could make ATP, right, which would be the output of this, if it's an electron gradient, right? And they said maybe these proteins are involved in something else. Maybe it's not the TCA cycle, but it seems kind of unlikely, right? Because they're all the right enzymes. Anyway, they do a lot of discussion about that. But then they ask, are these genes actually transcribed? And 6 of 8 of them are transcribed, but...
No, 2 of 8. 6 are not, right? I think it was 2 of 8.
6
I think 2 of 8 are in the particle. Proteins.
Oh, sorry. Okay, yeah, protein, sorry.
Yeah, 2 of them, they didn't find any transcripts. But the 6 that were transcribed were at the end of the reproductive cycle. At the end. Which, if you need energy, you need it early, right? It seems to me, I mean, looking at it as a human being, of course, I would want the energy to be made early. I don't know, maybe they're involved in assembly and not energy generation. I don't know. But that was weird to me. And then, yes, 2 of the 8 proteins are found in particles.
Maybe they help the cell as the cell is on its last legs during assembly.
Yeah. Finally, they take these genes and they express them in E. coli to see if they can get any enzymatic activity. They got proteins for some of them, but no enzymatic activity except for isocitrate dehydrogenase of 2 of these open reading frames. And it has kind of a low activity, but they argue that there's some activity and therefore it can't just be random that this enzyme is there. They wonder if it's an ancestral version of the enzymes, right? Maybe it's very old, and so maybe it had a different function way back when, right? So this is all very enigmatic, but still, it's interesting what this potential is, how it got there, and what these TCA cycle-like genes are.
Yeah, I come away thinking, okay, some fraction of these particles have an electrochemical gradient of some sort sufficient to suck up some mitotracker, all right?
Yeah.
I don't know what the nature of that electrochemical gradient is, and I don't really know how it got there. I also know that there's some weak homology to some TCA cycle genes, and I can certainly understand trying to make an association between that and the presence of this electrochemical gradient. All right? So it's interesting. And I was just looking up, when Ed and I sequenced a chunk of vaccinia 30 years ago, 40 years ago, 1980? Middle of 1980. The only, at that point, there wasn't much of anything in the database. So when we got this sequence, we looked at what homologies there were with anything in the database. And one thing popped out. It was a gene called D8 that had just a phenomenal homology to carbonic anhydrase, which controls pH
buffering
in the bloodstream.
In the blood, yeah.
Okay. And the weird thing about it, it had a big domain that was, no question, carbonic anhydrase, and a transmembrane domain. Okay. So what? Carbonic anhydrase anchored to the membrane of a virus. This makes no sense at all. Years and years and years later, it turns out that this thing is one of several virus attachment proteins. Somehow, it picked up carbonic anhydrase and somehow uses this to attach to cells. It has absolutely nothing to do with carbonic anhydrase, and in fact, it's enzymatically inactive in that capacity. So viruses do weird things that way. And I was just looking up, wondering, okay, did this virus like pick up a cluster of TCA cycle genes from somewhere and uses them for God knows what, or maybe nothing, right? We'll keep them around for a while, see if we can find a use for these things.
Yeah,
so I was looking up, and it turns out that in some organisms, at least, some of the TCA cycle genes are clustered. So you could theoretically pick up a chunk of a genome somewhere and pick up a few of these all at once.
That's a good point.
Yeah.
All
Yeah, that's a good
interesting
point.
stuff.
I mean, just because they look like TCA, they
Who
may
knows?
be doing something else or nothing at all.
Yeah, so I think that in terms of thinking about the electrochemical gradient and the link with TCA cycle, I would be really intrigued to know what they could see in terms of electron transport chain. But
Sure.
in support of what Rich is saying, these are viruses that are often studied, at least in amoeba, which are phagocytic organisms that spend a lot of time phagocytosing
Eating.
a lot of stuff. So they're probably picking up a lot, at least the host is probably picking up a lot of different DNA from a lot of places that could eventually end up in the virus and may or may not have fitness
Sure.
advantage.
Yeah, sure.
I actually sent this paper and some questions because I was really feeling very insecure about the relationship between the TCA cycle and oxphos and all that kind of stuff, because it's always been a mystery to me. So this has been a great education. But so I sent it to my most authentic biochemistry friend, and that's Ed Niles, okay, and said, "What's up with this?" And he didn't have much to say for me other than he's an old man and can't remember any of this stuff. But he did say, "Are these genes essential for the
Well,
virus?"
that's a good question.
Okay,
We don't know. Right?
and we don't know, and that's because you can't do genetics at this point with this virus.
Yeah.
You can't. The way you would do that experiment is knock out the gene in the virus and ask if the virus can still replicate, and you can't do that experiment. Okay, or at least you can't at this point. I suppose you could try some sort of microRNA thing. You know,
You could do microRNAs. You could do
Yeah.
CRISPRs. You don't
Right.
need to have, because this is a DNA virus, so you could knock it out with a CRISPR.
but that's a critical question.
But you need to have a plasmid encoding the genome to do a CRISPR knockout, right? So, it's too big, 2.5 million base pairs. You'd have to do it in several steps. But yeah, that's the essential question: do you need these things at all? And their last sentence is whether these results raise questions about whether Pandora viruses are still viruses. No, it doesn't. You haven't shown anything about. I mean, you find some kind of gradient. You find some enzymes that look like Krebs cycle enzymes. Well, you don't know if they're needed. So how can you say this questions whether it's a virus or not? No.
And
You
to
can't.
start out with, it was only 20%, I think, of the cells that you could even detect this
Yes, not
the
everyone.
membrane
Yeah.
potential. So it was like already 20%. And then the enzymatic
suspicious,
activity
right?
seems like background almost. So
Yeah, you kind of want to be like, guys, this was cool. You didn't have to try to give us a sales job.
yeah.
Yeah. Well, that's what they like to do with their giant
Yeah.
viruses always.
Yep,
That's what they
a
like to do.
common theme with this couple of groups in Marseille that have done all this
That's
stuff.
right.
Yeah.
No, I think Koonin has probably already weighed in on this. and I'm sure
Probably.
he would
probably out
not-
there somewhere.
not be happy about it. Because at the giant viruses meeting, they always argue. Koonin and these guys, they're always getting up and arguing.
So, audience out there, this is Critical Thinking Live. Okay?
Well, also, it's also us talking about these metabolisms that we don't talk about so much,
Right.
and I just love them. I love understanding what's going on.
Yeah. Yeah, I've actually learned a lot from this. This is great. It's always fun preparing for these TWiVs because I learn new stuff every time, and this one was particularly good in that regard.
All right, let's do some picks, folks. Angela, what do you have for us?
I chose 27 new exciting and blobby species discovered in the Peruvian rainforest. So obviously I love animals and I thought this was cool. And they had this one species of fish that they defined as the blob-headed fish. It's a type of catfish called Chaetostoma sp. Stoma obviously means mouth. And this thing, it's hideous. I mean, if you look at a picture of it, it almost looks like, oh, what's the name of that cartoon? I forget. It has like a huge dilated nose and they're very unclear. It's unclear as to why or what that function is. But one of the other things that I love, they have this little mammal, this little semi-aquatic mouse called it's Dapistomus sp. And this little guy in the Peruvian rainforest lives half of its life in the water. So it's like it has webbed toes and it's so cute. So this is just, they also define multiple other types of fish. Like I said, 27 new species, which I think is so wonderful because we think that we know, the creatures that are living on our planet. And we know so little even about the animals, let alone viruses and microbes. But the actual animals living on our planet are still there's still a lot of mysteries there. So I thought that was cool.
It's neat. Blobby.
It's a hideous fish. Yeah.
Rich, what do you have for us? Oh, I'm sorry, I skipped Brianne. Brianne, what do you have for us?
That's okay. So I have the planetarium show at the American Museum of Natural History in New York. They've opened this new planetarium show this month called Encounters in the Milky Way. I've mentioned it before because I had read about it, and I read about how when the scientists actually did the visualization, they discovered some new things about astrophysics just in trying to take data and put it into a planetarium show. And I thought that was really cool to read about. This week, I actually got to go to a couple behind the scenes events about this show. So I've now learned a tremendous amount about the show, and it's just amazing. I recommend it so highly to anyone who wants to go. The curator spoke a couple of times about it, and basically this came from a telescope in 2017 called Gaia that got a bunch of new data, and she described it as increasing the number of stars that we could see 15,000-fold.
Wow.
And so as a result of that, she wanted to make a visualization to kind of show that change to people. But it also allowed studying the motion of those for the first time and sort of getting to see them not as static stars, but moving. And so this show actually shows them in motion, shows that full number, and really kind of zooms from Earth all the way out. And there were just so many realizations I had about space that I hadn't realized before. It's really fantastic. I can't recommend it highly enough. And I was so inspired about the way that they were doing science communication and trying to think about ways to do like how would one do a zoom in the same way that they did this zoom out. And I've been super geeking out about science ever since I saw the show.
I'm hoping that this is something that can be exported to other planetaria around.
Me too. I wish.
Yeah, it's entirely possible. She also told me a little bit about one of the pieces of software they use, which is an open source software that NASA has to actually do some of the data visualization called OpenSpace. And so I've also done a little playing around with OpenSpace myself, which was one of the things that was used to make this.
Cool.
Did you go to see this on Wednesday?
I did go see it on Wednesday, and I saw some behind the scenes stuff as well.
Okay,
Yeah.
cool.
I love shows like that where you're sitting there and you're even just seeing anything. My sister, she talks to me about astrophysics, whatever. She's talking about space. And I always just sit there and I'm like, you realize our cosmic insignificance in the world.
Yeah. Well, and I had realized that a lot, but like one of the scenes in this, they actually show the tracks of every spacecraft that we've sent that has left the solar system,
cool.
how far they've gone compared to sort of how far there is.
Oh, that's great.
And you know,
so
like
cool.
one of them basically left when I was born. And so it's sort of like how far you could get in my life. And it's not very far.
Yeah, it's so
It's
cool.
so cool. Rich, what do you have for us?
So I have a Washington Post article, which I put the guest article link in the show notes. Unfortunately it's behind a paywall, but I'll try and help out with that. called "Where to Go for Accurate Up-to-Date Vaccine Information" by a woman, a doctor named Leanna S. Wen, who I linked her name as well to a Wikipedia article because she is a pretty remarkable individual who's trained as an emergency room doc, but has been active in a number of different public health endeavors over time and has been a consultant, et cetera. She was even, for a brief period of time, president of Planned Parenthood, for a brief period of time. At any rate, so she is, from what I can tell, the real deal. And in this article, she says, okay, things are pretty messed up here because the ACIP has been, that is the Advisory Committee on Immunization Practices, which we have discussed, has been dissolved, and in their place have been put people who are not appropriately qualified. And so where are we going to go for accurate information? And she lists a number of different things. And I will call those out so that those of you who can't get the article can at least know where they are. Jolene still doing timestamps for these episodes?
Yep,
okay. Jolene, heads up. Okay. Mark this as the list of alternative sources for vaccine information. The American Academy of Pediatrics has its own source, and it has access by its website. The American Medical Association, the American College of Obstetricians and Gynecologists, the National Foundation for Infectious Diseases, the Children's Hospital of Philadelphia Vaccine Education Center, that's Paul Offit's organization, the University of Minnesota's Vaccine Integrity Project, and the Straight Shot by the Center for Science in the Public Interest. So there are going to be, and there are already, sources out there for accurate information. She also says, don't forget about the CDC and the FDA, okay? Because it's not like everything that's going to be there is going to be compromised. But you have to do, if you're really interested in what's going on, you're going to have to do this due diligence. You're going to have to learn and practice critical thinking and go to these different sources and compare the information that you get and try and sort it out. Unfortunately, that shouldn't be the way it is. There ought to be a reliable central government source. But for the time being, that's unavailable. The one that really stands out to me that I've seen at least recently promoted is this University of Minnesota's Vaccine Integrity Project. Have we talked about that here before?
Yeah.
Okay.
Yeah, that's the one that Michael Osterholm kind of put together.
So some of these will probably rise to the surface as, and I would kind of hope that basically all these people would get their act together and combine their resources into one reliable site. But there will be reliable information out there.
Whatever it is doing their own thing. But they've done it since 1930, apparently. They always did their own recommendations. So they're still doing it. What is it? The American Pediatric Association or something like that. Yeah. Oops, he's frozen. Well, we got a good run out of him.
He lasted almost 2 hours.
Wow. I was just thinking, that's probably what jinxed them. I wonder if these are all going to be in little pieces now. Oh, boy.
I think it's my machine, and I will fix it.
Okay.
I think I know what's going on.
All right. Thank you, Rich. Now, in the same vein, I have an article from Science News, Science Magazine. New vaccine panelists have published Little on Vaccines by Martin Enserick and Meredith Wadman. And I like this because they have a graph of the publications on vaccines published by the fired members and the new members. And so, I mean, on the average, the fired members published 49 papers each on vaccines. And the new members have published an average of 11 papers, but 1, 2, 3, 4 have not published any, and most of it is by Martin Kulldorff and Cody Meissner. And Malone, of course, who used to work on mRNA before we had mRNA vaccines. But the graph is really visually striking. And the article goes on to talk about the loss of expertise and what some of these panelists, what we can expect from some of them. For example, Malone is a biochemist who, did some basic research that led to mRNA vaccines. He claimed that he invented it, which is wrong. So right there, we should have known that something was going on. Now he alleges that COVID vaccines caused 17 million excess deaths, which is a lie. They have not caused 17 million excess deaths. The data are there. You could look at it and know that it didn't do that. It's ridiculous. But he got a lot of attention by saying that. He went on Rogan and said all this crap and nobody challenged him. So he's one of the members. Great. Anyway, this is a good article. Check it out if you can get in there. Then we have 2 listener picks. One is from Charles. A pick from the Pipeline, Vaccine Advice Vandalized. So this is by, the Pipeline is written by, I forgot his name. Who writes the Pipeline?
It doesn't
writes?
say at the top.
Yeah, usually they have a...
Yeah, that's weird.
Derek Lowe.
Derek Lowe, thank you.
There you go.
So it's all about the trashing of the ACIP. And the best is the last paragraph. It says, the majority of the ACIP will now share most of RFK Jr.'s views. They will have meeting after meeting where they chip away at present vaccine recommendations. It will all be covered in banners of freedom and parental choice and doing your own research. But the result will be what Kennedy has been wanting for years, the undermining of the entire childhood vaccination schedule. This ignorant, willful man and his ignorant, willful president are going to have yet more blood on their hands because this demolition of the U.S. vaccination system, if accomplished, will absolutely lead to more illness, suffering, and death. This is one of the times I wish I believed in some sort of divine justice, some sort of karma, and some sort of hell. That's pretty strong words.
Pretty strong words.
He's not wrong.
He's not wrong.
Yeah, no.
He's not.
And somehow they'll still try to blame scientists, I'm sure, if children start dying, which hopefully they don't, but they will if they're not vaccinated, and
No, they will make up
and
lies
they'll make up
to
that
explain
it's our fault.
Yeah.
Somehow it's scientists' fault. That it's not their fault, of course. But
And we have a
the
pick from
surreal
Ken.
world.
He sends a link to the Golden Goose Award awardees. It lists scientific projects that sound unimportant, but ended up being very important. So I don't know if you remember, some of you may remember, William Proxmire was a senator who used to have the Golden Fleece Awards. He would look through NIH grants and pick ones out that he said were ridiculous. And so this is the Golden Goose Award because these all turned out to be really good. So that's a link of research on agrobacterium.
And on penguins, from poop to protection, satellite discoveries help save Antarctic penguins and advance wildlife monitoring. Yeah. Amazing.
Anyway, thank you for that. That's really good. How a lab incident led to better eye surgery for millions of people. So thank you for that. All right, that will do it for TWiV 1231. You can find the show notes at microbe.tv/TWiV. You can send us questions or comments to twiv@microbe.tv. And if you enjoy these programs, we hope you do, please consider supporting us. It's microbe.tv/contribute. Brianne Barker's at bioprofbarker.bsky.social. That's Blue Sky and also Drew University. Thanks, Brianne.
Thanks. I learned a lot.
Andrea Mingarelli is at McGill University. I was going to put you in a fish university, Gill University. McGill University, Immune Vet on Blue Sky. Thank you, Angela.
Thank you. I'm excited to see everyone at ASV, those of you that are coming.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Always a good time. Great to be here. I love this.
And Vincent Racaniello, you can find me at Microbe TV or on Blue Sky. Microbe TV is at microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.